the lectures are similar to what they were before, and so you can use the old sites to look ahead a bit, but they will change in some areas particularly. So just keep an eye on that. The lecture slides and lecture handouts are available on the course site, so you can click on those links in order to get the slides and a handout, which is a version of the slides. It's sort of formatted like this, where you can see, so it's easier to print out. It also has, for all of the figures, the code used to generate that figure, in case you're interested in how it was made, or if you want to make it yourself. Looking at sort of the intro questions, the range of backgrounds is quite broad. I mean, so the programming experience question covers quite a big range from limited to very experienced. And so what my idea was to have sort of the first few lectures do the visual or KNIME-based approach. And from what I could tell, no one had used KNIME before. Is that correct? Okay. And so we go over that just to understand the ideas, and then we would have, for the students who are more comfortable with programming, the opportunity to use IPython Notebook and some of those tools. Has anyone used that before? Good, okay. To do some of the analysis, because that makes it easier for you to do. And I think you should be able to see these after you fill out the survey, so you can get an idea of where you fit in the course. And the idea is that if you already have a ton of programming experience, it shouldn't be boring, but if you don't have any, you won't drown. And so the tools we use for the analysis are quite basic. So this is what KNIME looks like, and since no one's used it before, everyone's sort of on the same level. And I'll talk in this a little more detail in the later slides, but it's graphical, and so you drag and drop things on the screen, and it's fairly easy to use. Okay, so now getting to the actual slides of the course. So, as kind of an overview, so today won't be too technical in terms of the material that will be covered. It'll be more of going into the basics of the course so that everyone starts off at the same place. And the idea was to cover who are we, so who are the people who are behind the course, why are we teaching this, kind of what your background is or what's expected from you in this course, and what we hope you will get out of it. And then the idea of sort of why does this class even exist. So I'm sure, as most of you know, there are computer vision and other image processing classes at ETH, and why did we go through the effort to make this class if they already exist, and what are sort of the differences there. And then we'll talk a little bit about sort of what is an image, where do images come from, and sort of understanding that basis. Science and reproducibility, which I think today particularly are very, very important topics when you're doing research projects, and you should have an understanding of why this is important and the ability to do research that follows this sort of way. And then this idea of workflows. Okay, so in terms of who are we, so I'm the main lecturer for this course. There'll be some guest lectures who will come in to present on specific topics. I'm the CTO of an ETH spinoff that does big image analytics, which is sort of why I'm interested in this material in the first place. And then the lecture assistant for the course is Johannes. You should have gotten an email from him yesterday with a login and password for these machines. If you didn't get that email, somehow you're not registered right for the course, or your email address doesn't match up, and so you can talk to him to get an account so you can log into these computers. If you're an ETH student, you can use your standard login on these machines, but if you're not ETH, you have to use the login he's gives you. And two people can't use the same login. So if you try to use it twice, it will cause problems. And he's a PhD student in the X-ray microscopy group, and he took this course last year. So for sort of the other guest lectures, and this isn't all of them, this is just two, Anders Kastner, who will present next week, is at the Neutron facility, the Icon Beamline at the Neutron facility at Paul Scherrer Institute. So he does images with neutrons, which allows you to see through sort of heavy metals as if they were transparent. And so it's very interesting for looking at sort of engines that are running in real time or other sorts of things that are very difficult to image with other modalities. But the images you get out are very noisy and very messy. And so he's really an expert in how you can clean that up to make the images look better. The other guest lecturer that we'll have is Christian Dietz, and he's from the University of Constance. And he's one of the main people behind the KNIME tool and KNIME image processing, which is the tools we'll be using at the beginning of the lecture. And so he'll show you how they use that for sort of the cell imaging projects that they work on, sort of the light sheet and cell imaging projects that they work on. He's also sort of a good person to contact, and the person who will probably answer you if you go on the forums and ask questions about these things. So the idea of who are you? And so based on the people who've registered in years before and everything, it sort of ranges from biomedical engineers to physicists, chemists, and art historians. There's a huge range of skills, and so I don't know if this necessarily applies this year, but last year there were students who'd done sort of large-scale programming projects on their own, where they used very advanced topics and lots of optimization, to students who had never programmed anything before. I think both of them were able to get something out of the course, but of course your background's different and what you can achieve will be different if your skills are not at the same level. And so the idea is that the focus for this course will really be on making conceptual, or having a conceptual understanding for what topics are important. And so rather than focusing on implementation, it will focus on what are the right steps to take and why are you performing the steps that you're performing, rather than having sort of a lot of detail on for loops and optimization, which is really irrelevant for scientific problems. The point is to understand what you're doing and why it makes sense. And sort of what makes it more interesting, I would say, for the other students is that you'll have the opportunities to kind of create your own tools and make more advanced analyses where the standard analyses don't work. And so if you are at this higher level, understanding the basics will sort of be the first goal, and the second goal will be then to kind of take it beyond to solve your specific problem or to scale it up better. So in terms of the course expectations or how it will work, this course is sort of divided evenly between a lecture component and an exercise component. And they're sort of merged together in this one block, because I think the exercises are quite important for actually having a feeling for how to do these analyses and understanding sort of the more conceptual topics that are learned. That if you learn about segmentation and then take an image and segment it, you're much more likely to remember what you did than if you just learn about some sort of hypothetical topic. Yeah, and so they're optional. You don't have to do any of the exercises. They won't really be graded in that sense, but they're strongly recommended, and they help you to understand the material. And so, in my experience, examining the students who haven't done the exercises usually struggle with the exam, because I don't ask you what did it say on slide 5, I ask you to explain an idea and how it could be relevant. And so if you haven't collected any practical experience, it will be difficult to answer those kinds of questions. There's also this science project, which again is optional but strongly recommended. And so the idea of this project is to really take the topics learned in this course and apply it to a relative project or a relevant project for what you're doing. And so since many of you are PhD or master's students and have research projects of your own, I think it's quite helpful to, show first of all what other people are doing and get feedback from other people who might have interesting or new ideas for the kinds of analysis that you can do. This is also where having sort of the workflows is very helpful, because then you can show someone exactly what you did and they can easily modify it and understand which steps you take and ask questions about why you did that. And so everyone should be more or less at the same level for understanding and questioning sort of what procedures went on. And the idea is that you sort of, over the course, you analyze your own data, you build up little analyses for this, and then you present it for 5 or 10 minutes at the end and just say sort of what were your goals, what did you do, and sort of the justification for your steps. And I think it's interesting because you don't want to just hear from me or sort of the guest lecturers. It's also interesting to hear what the other students are doing, and sometimes you're working on much more advanced projects than we are, depending on what lab you come from. So with this course, there's lots of literature and useful references, and this is just a starting point. On the course website, there's a wiki. I think if you go to the view on GitHub, you can then get to the wiki for the course, where you have other sort of pages there, and you can feel free to make your own pages and add new lists of resources of books that are helpful or topics that are explained in more detail there, so that you can start to see it. This is just sort of the basics of what I've prepared for the course already, but feel free to add to it. It also lists other tools or other frameworks that might be useful for specific kinds of problems. But yeah, and so some of them are short articles that might be interesting to read that I'll reference over the course of it, and other of them are much longer books which are helpful for understanding a topic in more detail if that's something of specific interest to you. Cool. So kind of the motivation for the course is that, and this is slightly exaggerated, but that basically, you know, when you have a science project, that you go from having maybe a bunch of bones you measure in, you know, from mice in 1 or 2 groups, and then you have kind of this black box of things that happen, and then you have some result in Blick am Abend that says science says ice cream is good for bones. And the point of this course is that all of these steps that happen in between, we won't really focus on the imaging itself, but once those images are taken, how do you go to that final result? So how do you go from having just a ton of data to being able actually to test a hypothesis and show, for example, that ice cream is good for bones, and then be confident that you've done that correctly and that other people are able to get the same results that you are. And so it's really sort of a scientific method course focused on image analysis. And I'll talk about sort of why images are a little bit special in that way in some of the later slides. And so the idea here is that you really understand the what, why, and how from the moment an image is produced to the final result that you get, and that you learn a lot about the scalability, and so that you do things in a way that, if you have 10 times or 100 times as many images, that you're excited by that, because that means your statistics will be much better and you'll be much more confident in your result. And what we've typically seen is that if you have 10 times as many images, it means you have 10 times as much work, and so you're not excited about it. And so if we can make this a good thing, that will ultimately help improve sort of the quality and the reliability of the results that we have, so that reproducibility won't be such a major issue in science, or that would be the vision of this class. So, kind of the other specific topics of why this class exists, and I'm sure many of you are familiar with some of these things already, but sort of the detectors that you're using in imaging, particularly at X-ray and optical sources, are constantly improving and getting larger and faster. And so that means you can measure right now at the Tomcat beamline at the Swiss light source, which is the example I know best, 8 gigabytes a second of data. And so if you're trying to analyze this with standard or existing techniques, if you have a nice machine with maybe 32 gigabytes of memory, it's full in 4 seconds. And so the standard approach isn't going to work very well for data rates that are that fast. And so understanding how you can scale that up or handle larger volumes of images is really important. A single camera, where one of these single cameras can produce more information a day than Facebook. And Facebook has hundreds, if not thousands, of PhDs in computer science developing the most advanced tools for them. How can you possibly hope to keep up with that as a scientist, or to make something meaningful out of that? There's also other experiments where sort of diffraction patterns are collected at 30 gigabytes a second, beamline at the Soleil, which is the synchrotron they have in Paris, measures about 10 terabytes of data a day on a regular basis. And so this is sort of so much information that you're really overwhelmed by the processing. And then you see even sort of in your personal life, you know, if you have a GoPro 4 Black, you can take 4K video at 30 frames a second. And if you wanted to count cars or identify faces in all of these videos, this would overwhelm your computer very quickly. Cool. And so what I've seen, or we've seen, sort of the experimental side of things, is that the experiment's time breakdown has really shifted. And so if we look in 2000, for imaging experiments, there was sort of a fairly, maybe not completely even, but fairly even split between designing an experiment, doing measurements, managing the data, and then doing the analysis. And so you measured data in a week, and maybe you spent a month analyzing it, but it wasn't so much different. And what we start to see now, and even more in the future, is that you can measure data very, very quickly. And so you end up measuring data in 3 seconds or 2 minutes that you spend 3 years analyzing. And so if you're trying to really understand this data well and keep up with your new sources, then you need better tools for analyzing it, because you can't have this mismatch between how much you can analyze and how much you can measure. Cool. We've also seen sort of kind of a saturation in output, and so even though the amount of data we can produce, the amount of information we're getting out of it, hasn't increased. And so a lot more data isn't helpful unless you really understand what it's coming from and how to interpret it well. And if you sort of think about how much is a terabyte of data, really, if you looked at one image like this, which is 1,000 by 1,000, every second, it would take you 139 hours to look through a terabyte of data. And so if you're looking at an image like this every second, you obviously can't be doing a very complicated analysis on it. You're just looking at it and seeing if you see a pattern or something. And so if you wanted to keep up with this 8 gigabyte a second camera, you'd need 4,000 people looking at images like this 1 a second. So that's clearly not a realistic approach, and you need to sort of employ other techniques in order to keep up with these data rates. So now, in terms of kind of what analysis you want to do with these images, you see in biology in particular that there's a lot of images that people measure or look at, or pathology, where your goal is to sort of analyze cells on a slide. So this is a bone slice, and the idea is that you want to count how many cells are on this bone slice. And if you have one slice, then it's fairly easy to count them yourself, and it's not a problem. If you then count them and write the number down, and go to your boss and he asks you to ignore the ones that are too big or shaped strangely, then you have to go back to this slice and count them all again. If he then says, are there more on the right or the left, then you have to go back and count it all again. And if there, are the ones on the right bigger than the ones on the left, or other questions like that, every single time there's a new question, and this is constant in biology. I mean, these are all hypotheses that you're trying to test. You have to go and count it again. And so this is incredibly time-consuming and inflexible. And so with one slice, maybe it's reasonable to do this. But once you start to have more data, it gets much more difficult. And so if you have 96 more samples, then it's much worse. And then if these samples are 3D instead of 2D, and you have 2,000 slices instead of just one, you're dealing with a huge amount of data to try to analyze. And that simply isn't feasible. And then when you scale up to even bigger projects where you have 1,090 samples, it's then not even feasible. I mean, the time it would take a person to count all the cells in all of these samples is, I think, around 100 years. And so you have to employ automatic approaches in order to deal with data sets this large. And data sets this large, particularly in biology, are important for looking at things like genomics. That if you're trying to understand what effect a single gene has, you need a huge number of samples because there's so much variability that you won't see small changes without having this kind of data. One of the other major problems that you see is that If you use sort of qualitative approaches, which are very common in image analysis today, it's helpful. I mean, a lot of human eyes are capable of seeing structures that are difficult to implement for computers, but it makes it very difficult to compare a large number of samples. And so if you're trying to look at these cells, for example, these sort of small ones, if you want to describe how well aligned they are, if you do this qualitatively and you say kind of aligned, well aligned, not really aligned, that might work for 3 samples. But how can you compare the one on the right to the one on the left? Or how can you measure if something made them slightly more aligned? And so it really takes away a huge amount of the ability for you to compare samples, and particularly when you're looking at hundreds, to have a reliable estimate for this. Because maybe you thought this one was really well aligned, but someone else looked at it and said it was not aligned because these things are going in the other direction. And then you have sort of a conflict. And without having everyone with the exact same definition and exact same understanding of what alignment means, you're going to have this problem. So I don't know if this... That link doesn't work now. Anyways, so this is... I don't know if I have that video. So, the video that's supposed to be there. That's weird. Oh, that's one. The video that's supposed to be there is this video. And so this is again from one of the 3D experiments we've done. And this is then a liquid foam. And so all of these holes that you see are bubbles, and this yellow material is then sort of the water. And that's been segmented into the image. And this is the result of the experiment that you get. And so now if you look at that image or look at that movie and try to measure this information out of it, you know, how many bubbles are here? That's a very difficult question to answer with a movie like that. I mean, you could go through and try to count all of the bubbles, but it's very difficult to see them in 3D, and it would take you a long time to go through the whole image in every frame to see that. Then if you have to answer questions like how fast are they moving, then you sort of have to find a point and measure how quickly it moves. And then if you have sort of these other questions, you know, do bigger bubbles move faster? Are they moving slower near the edges? Or what's very interesting for sort of mechanical and material sciences is are they rearranging? And so if they're rearranging, this changes their sort of mechanical and flow properties substantially, versus if they're sort of flowing in a static block or plug, is what they usually call it. And so if you want to have information like that from a video like that, you can't do analysis manually. It's simply too much data, and there are things that are way too difficult to try to see in a single image, or in sort of an image with your eye. One of the other things that has sort of rapidly changed recently is that sort of, you know, I'm sure all of you are familiar with Moore's law, that the number of transistors doubles every 18 months, which is true. But recently, and this is sort of since 2000, the speed of the computers themselves. So this Moore's law has held true more or less, but the speed that the computers operate at has remained constant. And so many of you have also seen this in your cell phones, that your cell phones might have 4 or 8 cores, but the cores themselves aren't faster. And so what this means is that when you're trying to do processing tasks, you need to be able to divide them up into little chunks on many different cores efficiently, or in many different processors or machines efficiently. And that sort of up until 2000, the strategy could be, if your algorithm isn't fast enough, you can wait a year and the computers next year will be so much faster that you don't have to spend time, you know, optimizing or parallelizing it. But now what's become sort of the case is that your algorithms won't get faster on their own. There has to be some way to parallelize them. And so this has been a huge problem. and has really forced a lot of changes in the way people think about these sort of approaches, because this parallelization is not easy. And particularly if you're working at a very low level, it can be very complicated to implement yourself. And so one of the things that we show in the sort of workflow and other approaches that we have is that if you focus on a much higher level, where you just sort of link components together, then you don't have to worry about the parallelization or the optimization. That handles other tools in the background. And so with tools like KNIME or TensorFlow or MATLAB, when you're doing these tasks and you perform large operations, it automatically breaks those operations up into little pieces. And so then it can perform it very quickly. When you're using tools like C++ or sort of coding things yourself, this you need to manage yourself, and it can be very complicated and can make very unreliable results. And we'll talk about that in one of the later lectures. One of the other things that sort of changed recently is this idea of cloud computing. I mean, so at universities, there's always been cluster computing, but more recently, this has become available to really everyone. And there's companies like Amazon, IBM, Google, Microsoft, which offer clouds. And so what this means is that you have huge clusters of computers that you can borrow very cheaply whenever you want. And so when you look at sort of